Hello everyone, I'm very excited to be here to talk about my work on developing holistic predictive models of cortical responses to naturalistic stimuli. This talk is going to focus on computational models that can predict how the brain will respond when presented with naturalistic audiovisual stimuli. So here's the setup. Subjects passively watch movies inside a scanner while their brain responses are being recorded with functional MRI. In this work, we model these brain responses using deep neural networks to understand how the brain processes this incoming sensory information with two goals in mind. First, we want to build better models that capture brain activity more stringently than existing approaches. Second, we also want to probe these models to elevate our understanding of the brain. Here is the illustration of our approach. The simplest model we use is a unimodal model that predicts neural responses using single snapshots of the sensory signals, either input images or audio signals represented in the form of a spectrogram and fed through a hierarchical convolutional neural network. We also train 20-sec versions of these models that extract the same features from a sequence of 20 input images or spectrograms but uses a recurrent network to dynamically recompress the input sequences. Finally, we have a multimodal version of these models that use auditory and visual features jointly to make predictions. By varying the amount and type of information used by different encoding models and comparing the resulting prediction performance, we can characterize how neural resources are distributed across the cortex for processing naturalistic video sequences. In our first set of results, we computed the mean prediction accuracy in 5 groups of regions. The first 2 groups, comprising auditory and visual regions separately, do not show marked improvements from using multisensory inputs. The performance boost with multisensory and longer timescale inputs is more pronounced in the remaining 3 groups, which are not preferentially associated with a single modality and are involved in higher order processing of sensory information, namely known multisensory sites, language processing areas, and regions situated in the frontal cortex. Another interesting observation we make is regarding the timescales. Using prediction performance alone as a metric, we can discern a temporal hierarchy within the auditory cortex, as there is a small contribution of longer timescale inputs on prediction accuracy within early auditory areas, and a much larger improvement is seen for the higher auditory regions. Following a similar approach, we observe that the performance gap is higher between dorsal stream areas, whereas no difference is seen across the ventral vision areas, consistent with the known association of the dorsal stream with action-oriented behaviors and ventral stream with object recognition. Next, we want to investigate the possibility of using encoding models as neural activity synthesizers. Given that we have a predictive model linking stimulus and brain activity, we can feed it stimuli from alternative paradigms such as typical stimuli from task fMRI experiments and test how well these models transfer to this new domain. In this experiment, we predict the brain activity for different images and then use the predicted activations to generate the contrasts for 2 well-studied visual categories, namely faces and places. And we find these models, despite having only seen naturalistic image data during training, can indeed generalize remarkably to these synthetic images and replicate the contrast maps for processing faces and scenes. To conclude, using just 6 encoding models trained only on nearly half an hour of naturalistic data per subject, we can replicate findings from a large number of prior studies on sensory processing. Specifically, a single naturalistic fMRI experiment helped us characterize the functional modules involved in facial and scene processing. It helped us distinguish the temporal hierarchy and reproduce the multimodal architecture of the brain. Hopefully, this convinces you that the interplay of naturalistic neuroimaging and predictive models can be very exciting in neuroscience.